Part 3. Introduction of sodium lauryl sulfate, sodium dodecyl sulfate, and enzymes to detergents in 1960. We see it as SDS and SLS. And 1960 is a milestone time because these additives were added to detergents, and the detergent industry made a big change by making all the clothes very clean, and the laundry was quite successful for the cleaning effect. And sodium lauryl sulfate in the beginning was added around 20%. Now it's decreased to 10%. And it is in many detergents, there is 10% sodium lauryl sulfate. In shampoos and household cleaners, you can see 5 to 10% sodium lauryl sulfate. And in some toothpaste, after 2000, we are seeing that sodium lauryl sulfate is added in the range of 3%. And it can be extracted from house dust, pillows, bed sheets. It is toxic to cells, and we can see it in organoids, organoid chips. And the dose of toxicity is 1 to 50,000 times. And there's in vivo studies, and the toxic threshold is 0.002%, whereas it is included in these detergents and toothpaste around 3 to 10%. So for this area, please read the papers of Hideaki Morita, Arturo Rinaldi, Mu Xian, Ming Wang, Yasutoka Mitamura, Alfred Doyle, and Benjamin Wright. So here we have the 50,000 times diluted sodium lauryl sulfate or laundry detergent that affects the epithelial barrier. What we are seeing in the transcriptome is that there is upregulated lipid metabolism, oxidative stress, endoplasmic reticulum stress, cell survival signals, and interleukin-33 and alarmin. What is downregulated is cell adhesion, extracellular matrix organization, wound healing, and DNA repair. So the interpretation of this finding is a chronic wound that tries to survive the cells, and it's a carcinogenic environment because of endoplasmic reticulum stress and DNA repair defects. So here is a very exciting study coming from Hideaki Morita and his group. And here we can see that only 4 drops of a detergent, or in the same study we can see sodium lauryl sulfate also, added to the nose of mouse, goes to respiratory system and cause an eosinophilic bronchitis. And the peribronchial, very strong eosinophilic asthma-like model. And we can see total leukocytes increase in the lungs. Eosinophils shows a very significant increase, and there is some neutrophils in addition. And here we see IL-5 reporter mouse, which shows that mainly type 2 innate lymphoid cells are the main source of type 2 cytokines, making interleukin-5 in around 44.4% after detergent exposure, and this is the control. And basophils and T cells have no effect. Then we looked at what stimulates the type 2 innate lymphoid cells, and here we can see that detergent-exposed lungs have a lot of interleukin-33 production. And here we have interleukin-33 knockout mouse and wild-type mouse, and we can see that the total leukocytes do not change, but eosinophilia is increasing with the wild-type mouse, which decreases to normal levels in interleukin-33 knockout mouse. So the story is interleukin-33 coming from the epithelium after detergent exposure activates the type 2 innate lymphoid cells, and this makes eosinophilia. And this is an interesting study also coming from the group of Hideaki Morita. And here we see that ovalbumin-specific IgE is induced in detergent-exposed mouse. And this is a very interesting finding. And if we have the antigen alone, there is very little IgE. But if there is a detergent exposure at the same time, then we can see that IgE is going very high. And this is one of the first findings showing the induction of IgE with an environmental exposure, which can be the reason for the development of allergic diseases. We are using ex vivo human skin models coming from the group of Nicolas Gaudenzia, and we have made several papers. And we are also using an electric impedance spectroscopy, which is developed by our group. And this is mainly used to detect skin barrier. And with one touch, within 2 seconds, you can detect the skin barrier. And we have published several nice papers on this. And we have used here in this model, by using these 2 devices and by exposing to 2 bacteria, 2 very healthy probiotics, Bifidobacterium breve and Lactobacillus reuteri. And we can see that by extreme gradient boosting, XGBoost regression model, it's a machine learning model, we identified most critical genes associated with Bifidobacterium brevis induced barrier enhancement. They include OL2, keratin, and ETV4, LAT-S2, Notch, and MHY15, and cyclin-dependent kinase, and Claudin 10, a barrier molecule. So we are now in the way to identify what makes strong epithelial barriers and how can we modify these genes. And I would like to mention this paper. It's about development of eosinophilic esophagitis. Eosinophilic esophagitis was first described by Hans-Uwe Simon and Alex Straumann in our institute. Actually, the paper is coming from CIAF. And in this paper, they show the first series. There was single cases mentioned before this. And in this study, Alfred Doyle and Benjamin Wright showed that SDS, sodium dodecyl sulfate, which is the main culprit in the detergent and cleaners and also several cosmetics, is stimulating IL-33 production from esophageal epithelial cells, and we see very high increased IL-33. And this is SDS exposed esophageal epithelium and esophageal tissue. You can see a strong cellular infiltration here and thickening of the mucosa, and there's a lot of eosinophils increasing and infiltration of eosinophils. And this is for comparison, a normal esophagus. And we can see that in this paper, exposure to SDS causes decreased epithelial barrier integrity, epithelitis with increased IL-33 production, eosinophilia, and inflammation, abscess formation, and CD4 positive T cell infiltration and remodeling in the esophagus. And this is STS and SLS, sodium dodecyl sulfate, is the main culprit that comes from the toothpaste and household cleaners to make eosinophilic esophagitis. So what are our suggestions? Now, a group of doctors are working to find evidence and to suggest to patients to avoid these substances that are causing allergic diseases and autoimmune diseases. And we have SLS, STS and enzymes of detergents, cosmetics, and cleaning substances. And here we are suggesting to use an extra rinse and a small amount of detergent and opt for liquid detergents instead of powder. Choose SLS-free products. Read ingredient labels. Use natural or organic products to limit the use of foaming products, because the foaming products are changing the surface tension and then they are opening the epithelial barriers and initiating inflammation. Avoid prolonged contact. Usually less than 1 minute contact can be tolerated, but when the contact is long, we have data with 1 minute and also 5 minutes, the inflammation becomes very significant. And be cautious with heat, because heat also opens the epithelial barrier. Hot water opens the epithelial barriers. Together with these toxic substances containing cosmetics and detergents, the damage increases. You can look at the website epithelialbarriertheory.com, and there's a very nice app, Yuka, and this app describes almost all the substances. There's more than 2.5 million substances mentioned in the Yuka app. And you can identify whether the cosmetic or the substance or the food you are using is damaging the tissues, and together with the literature.